Good evening, everyone. Can you hear me? Okay. Welcome, all of you, to this, what proves, well, I know it's going to prove to be an amazing talk. I am Gifty Akowa-Dunvo. I'm the Vice Provost for Global Initiatives. I've been at URI now for about 7 months, and every day is exciting. So this is one of those exciting days. I'm not going to be too long here at the podium. I wanted to take the opportunity to introduce Professor Yalda Shahriari, who is an assistant professor of biomedical engineering in electrical, computer, and biomedical department here at the University of Rhode Island. Dr. Shahriari did her postdoc studies at the University of California, San Francisco, UCSF, and she earned her PhD and MSc in biomedical engineering from Old Dominion University of Virginia and Iran University of Science and Technology, respectively. Her research interests include neural engineering and computational neuroscience, biomedical signal processing, brain-computer interfaces, BCI, machine learning algorithms, and health unit data analysis. Her recent projects include investigating the brain connectivity patterns of patients with Parkinson's disease, PD, during deep brain stimulation, DBS, developing hybrid BCI systems for patients with amyotrophic lateral sclerosis, also known as ALS, brain synchrony and oscillatory analysis of mice models of schizophrenia, and decoding movement-related cortical activity in patients with epilepsy. Her main research is focused on a better understanding of brain functions using the cutting edge of neural engineering and cognitive neuroscience techniques, with the goal of developing translational medicine and ultimately improving the quality of lives of patients with neurological impairments. And with this, I'd like to introduce you to Professor Shahriari, who will introduce our guest. Thank you.
Thank you. Thank you. Hello, everybody. Thank you for coming. I would like to say a special thank to Provost and Vice President and Dean Wright and Haibohi, our department chair, ECB department chair. We are excited to have you here, and of course, Dr. Cohen. We are very excited about his talk. So I give a brief introduction about Dr. Cohen, and then we will have Dr. Cohen here. Dr. Michael X Cohen is an assistant professor at Radboud University Medical Center, Nijmegen. Sorry about the pronunciation, so it's a little bit difficult, but it's Nijmegen, Netherlands. I don't know if I pronounced right, but Netherlands, and at the Donders Institute for Neuroscience. His main research interests are neuroscience with a specific focus on neural oscillatory analysis that identifies signatures of cognition, including error monitoring and correction, as well as more fundamental studies of neural circuitry underlying synchronization. Dr. Cohen completed his PhD studies at the University of California, Davis in 2007. His PhD project was mainly focused on computational modeling of reinforced learning, reward anticipation, and memory. Then he joined the Clinic of Epilepsy at the University of Bonn, Germany, as a research scientist. Dr. Cohen's research as a research scientist shifted towards identifying patterns of neural oscillations and synchronization in patients with epilepsy and understanding deep brain stimulation mechanisms in patients with major depression and Parkinson's disease. Dr. Cohen then completed his postdoctoral studies at the University of Arizona, where he explored computational models of cortical basal ganglia interactions and EEG time frequency analysis. Afterwards, he joined the University of Amsterdam as a Amsterdam psychology department as a research scientist in 2009. During this period, he contributed to several novel discoveries about the human neural signatures of cognitive control processes and developed analysis methods for large-scale multi-dimensional neural data sets. Two of his famous books include Analyzing Neural Time Series Data and MATLAB for Brain and Cognitive Scientists. These books are highly popular nationally and internationally and are currently being taught by many universities to undergrad and graduate students. Dr. Cohen has over $3 million grant from various resources. He's also fluent in English and Dutch and a well-versed public lecture speaker. So please clap for Dr.
All right. Thank you, Yalda, for that kind introduction, and thank you for bringing me out here. And arranging the whole week, and thanks to everyone else who was involved in organizing this. I hope you don't think it was a mistake to bring me out here. You'll find out in an hour from now if you think it was a mistake. It's okay if you think it was a mistake. People make mistakes. Some people make more mistakes than other people. And unfortunately, I think I'm going to have to include this slide in my talk for the next 10 years. Anyway, I would like you to think for a moment about the last time that you made a mistake. Maybe you're thinking that you don't make mistakes, or you haven't made a mistake for years. And if so, then I encourage you to consult with your significant other or boss and ask them to confirm that you haven't made a mistake in a long time. Or maybe the last time you almost made a mistake, but you caught yourself at the last minute. Now, when you think about these kinds of questions, you might be thinking about sort of major mistakes, like did I choose the right career path or the right major in college? Did I pick the right person to be with? Did I vote for the right presidential candidate? These kinds of big decisions where maybe you felt like you made a mistake. But mistakes actually happen quite often. So big mistakes hopefully don't happen too often. Smaller, more sort of less consequential mistakes happen much more frequently. Mistakes are really universal. They cut across age groups, they cut across cultures, and they even cut across species. And this is probably, one of the sort of most enjoyable pastimes of modern technological life is looking at pictures of mistakes, particularly by animals. So here's some data showing that we make small mistakes quite often. So this is data from looking at keyboard typing, so naturalistic keyboard typing. And here you see the proportion or percentage of errors, so mistakes that people make while typing. And these are uncorrected mistakes. So it's about 0.5% of keystrokes are errors. And given the number of words, the average number of letters per word and the average number of words per minute, you make mistakes that are uncorrected, once a minute, once every few minutes. This is, by the way, data from 136 million keystrokes. This is a really large-scale study. So here's some data from my lab from several years ago. What we did here, what we have done quite often, is measure EMG. So this is the electrical activity of the muscles that the research participants are using to indicate responses on a mouse, for example, or on a keyboard. So here you see, so there's a stimulus, there's a picture that comes on the computer screen, and they have to press the left or the right button. And then here's when the button press was actually recorded by the computer system. And here you see the muscle, so it starts to twitch as the research participants are moving their thumb down towards the button. Now here you see an interesting trial. These are the EMG traces from two hands. Now the black trace here corresponds to the correct response, and the gray line corresponds to the muscle of the incorrect response. So this subject, this person, actually made the correct response on this trial, but a few hundred milliseconds, so a fraction of a second before they made the correct response, they actually twitched the finger of the incorrect response. Right? So they actually started making an error, and they somehow managed to correct themselves at the last moment. So this is called a partial error, and then we call the whole trial a mixed correct trial. And then if you look here, it's around 10% of of trials that people are making these partial errors. So again, this corresponds to a couple of partial errors every minute, so quite often. Now it turns out that people are not consciously aware of most of these partial errors, and that comes from a study that was not done in my group, done by a different group. Here's the reference up here. So I'm sorry, it's a lot of it's a big table full of numbers. These are the key columns. Essentially, what they did is train the research participants to know what a partial error is. So they showed them data like this and explained what a partial error means, and then asked the research participants to report on each trial whether they made or whether they were aware of making a partial error. So here you see the number of partial errors that were made. So it's in the hundreds over the course of about an hour. And this is their detection performance. And in particular, what you should notice is the high rate of misses here. So that means that the majority of these partial errors they evade conscious awareness. So people are not even consciously aware. Even when you train people about these partial errors, they're still not able to consciously access that they made their brain sort of activated the incorrect response that got all the way down, this incorrect signal got all the way down the spinal cord into the muscle. The muscle started twitching, and then they pressed the correct response. Most of the time, you have no idea that this is going on. So we make quite a lot of mistakes all the time, probably hundreds of mistakes every day, or near mistakes every day. So this is something that started troubling me a lot, like around 2000, I think 12 or 13, I started to get really bothered by this question and thinking about why do we make so many mistakes? We are supposed to be like the pinnacle of intellectual development on planet Earth, which is not to say that we're done evolving, but we are by far the most intelligent species on planet Earth, which means maybe the most intelligent species in the galaxy, for all, nobody knows. And yet we make so many mistakes that it felt really difficult to reconcile for me. In particular, when you think about how old the nervous system is from an evolutionary perspective, the earliest precursors of something that would ultimately form into a nervous system are around 3.5 billion years old. So that's really old. Almost as old as life itself. And what you could call a brain, that's maybe around 500 million years ago, which is a really long time ago. That was like before Twitter, things like that. So still hundreds of millions of years of evolution, and we still make mistakes all the time, hundreds of times a day. So so how to reconcile our intelligence with the frequency of making mistakes? And the only conclusion that I think is logical to draw from this set of findings is that the fact that we make so many mistakes is not itself a mistake. It's that we are designed, your brain is designed by hundreds of millions of years of evolution specifically to make mistakes. Nature wants you to make mistakes. And why is that? Why is it good to make mistakes? Why should we have a brain that's designed specifically to make mistakes? I think that part is, fairly, I think we would all come to the same conclusion, which is that mistakes are really the best way to learn. That's not always stressed in modern culture, and particularly in fields that become more competitive. But of course, everyone really knows that you learn more from getting things wrong. Of course, you have to learn from your mistakes in order for them to be useful. But that's a separate topic. So I'm going to focus a bit more on why we make mistakes, how we make mistakes, what are the brain signatures of making mistakes, and what we've learned about the brain from studying the brain's response to making mistakes. Okay, so how do we make mistakes? What is the mechanism of errors, of making mistakes? There could be many. I'm going to focus on two possibilities. One is noise. So the brain is actually pretty noisy, and noise is pervasive in the nervous system, all the way from the lowest levels of the nervous system up to the highest levels of behavior. And here you see an example of recording from a neuron, so a brain cell, that was isolated from the rest of the brain. So it's sort of in an isolated environment. It's taken out of the brain context. And what the researchers did was inject into this neuron a current, like a step function current. Couldn't be simpler than that. And here on this plot, you see the timing of the action potentials of the neuron, so the firing of the neuron. Each dot corresponds to an action potential emitted by the neuron over time. And then the y-axis here is trials, so they repeated the experiment 500 times. So you see that in the beginning, there is this relatively reliable timing of the response. But this neuron becomes more and more noisy over time. So it becomes stochastic, it becomes unpredictable. Its timing is hard to predict, even though the neuron is exactly the same, the stimulus, the input is exactly the same. Here you see another case. So this is with pseudo-generated or pseudo-random noise. This is slightly more biologically realistic. But still, the neuron eventually becomes more and more stochastic over time. And this is a pretty short period of time. This is just 1 second here. So you take a single brain cell, you isolate a brain cell, and it's still going to be quite noisy. In fact, so this is taken from a review paper published about a decade ago. They were describing here that noise is present in the nervous system at every scale. So every spatial scale of the nervous system that you look at, you're going to find stochastic responses or unpredictable responses. All the way down to ion channels and all the way up to behavior and motor neurons. All right, so that's one mechanism, one reason why we make mistakes. There's just noise, the brain is noisy, so that's going to cause us to make different decisions or take different actions. There's another possibility, which is this trade-off between exploration and exploitation. These are two strategies that are used for foraging. And so exploration is basically the strategy where you're constantly looking around for a better deal. You think that there's some better option out there. And exploitation is the strategy where you stick to what you know what you like, so you keep choosing that option. So these have pros and cons, of course. The cons are that if you're exploring too much, you might actually be missing the best deal. And if you're exploiting too much, so you always go back to the same restaurant and you always order exactly the same thing, then maybe you're missing a better deal or a better restaurant option. So it turns out these two strategies are associated with different networks in the brain. And in particular, this more exploratory strategy, which can lead to more errors, leads to more mistakes, is associated with a network that includes a region of the brain called the dorsal medial prefrontal cortex. And that also happens to be the region that we find that is most active for signatures of errors. So there's something about there's some overlap of the networks that are involved in exploring your environment with the networks that are involved in recognizing that you made a mistake or that you're about to make a mistake. There's a related concept called the matching law. And it's characterized mathematically by a deceptively simple equation, which basically just says that the rate at which you select a particular option is proportional to the rate at which that option reinforces your behavior or gives a reward. So if you're not too comfortable looking at equations, it's easier to explain the matching law in the behavior of pigeons, which is where it was originally studied. And so basically in this experiment, they put pigeons into like a box with a couple of wells in the box. And the pigeons just learned over time through trial and error that there was food pellets, there were rewards in some of these wells. So then it turns out if you reward option A, key A, 70% of the time, the pigeons will select that option 70% of the time. And if you reward option A, 15% of the time, the pigeons will choose that option 15% of the time. Now it turns out, so this is the matching law, basically. The rate at which you respond is proportional, or in this case, equal to the rate at which that option reinforces or gives a reward. This is actually a funny behavior, because if some behavior gives a reward 70% of the time, the actual optimal strategy, the best strategy, is really just to pick that behavior 100% of the time. You don't need to explore, you don't need to worry about making mistakes. You just pick that option all the time. You're not going to get rewarded all the time, you're still going to get rewarded 70% of the time. But you should pick that option 100% of the time. But okay, pigeons are stupid, right? Pigeons are not the pinnacle of intellectual development on planet Earth. Or maybe they are, they just have convinced us that they're not. So it turns out the matching law has been observed in many, many species, every species in which it's been investigated, including bees and including humans. So here is some data where we also, so the research participants had to pick just a stimulus on the left side of the screen or the right side of the screen. And one option would reward, let's say, 75% of the time, and then it would switch, and then it would reward 25% of the time. And again, the really, the best lazy strategy is when you figure out that this rewards more often, then you only pick this one, right? There's no reason to pick the other one until it stops rewarding. But that's not what people do. Okay, so so why is this the right option? Because the environment changes, so you don't want to just exploit your environment constantly. You need to do some exploration, but that also means you're more likely to make mistakes. You're more likely to choose the wrong option. Okay, so when I asked you when you make, what was your last mistake, probably you were thinking about big mistakes. You forgot to set your alarm and then you missed your exam or something like that. These are the kinds of mistakes that we are interested in understanding, but it's hard to study these kinds of mistakes. They don't happen very frequently, and some mistakes like marrying the wrong person, we can't really manipulate that in the lab. So I want to tell you a little bit about how we study making mistakes in the lab, and a little bit about how we study brain activity during when people are making mistakes. So I'll highlight, so there's 3 commonly used tasks. I think I'll skip the Simon task. You might be familiar with this task. It's called the Stroop task. And in the Stroop task, you as the research participant are presented words that are printed in different colors. And your objective, the instruction, is to read as fast as you possibly can, not the word itself, but the color that the word is printed in. So here you say blue, here you say, and here you say, okay. Not so bad, right? Because you had like 30 seconds to prepare. But if there's time pressure and these words are popping up one after the other on the screen, you have to respond quickly. You can imagine that people say green or yellow, right? So people make a lot of mistakes on these kinds of trials. Even when you don't make a mistake, even when you say the correct response, you still, you kind of trip a little bit on your response. This generates what we call response conflict. So there's a conflict between competing responses. There's a bottom-up response, which is your tendency to read a word when you see it, versus the top-down sort of task-instructed response, which is to say the word say the color that the word is printed in. I never make mistakes. All right, so more formally, we call these congruent trials and incongruent trials. So the famous Stroop task, which means syrup, by the way, in Dutch. So it's So Simon task, I'll skip just to make things easier. So then there's another one called a flankers task, and here you would see an array of arrows appearing on the computer screen. And your goal is to respond according to the direction of the central arrow. So here you would press the right button, here you would press the left button, thank you. And then there's these flanking arrows that you're supposed to ignore. And again, when you're sitting here and you see this for many seconds, there's really no response conflict, or it gets resolved so quickly that there's no real signature or response conflict. But you can imagine that if this is presented on a computer screen for a fraction of a second, and you have to respond as quickly as possible, otherwise we're going to give you an electric shock. I'm just kidding, we don't actually do that. But you're encouraged to respond quickly, and then people they will make more mistakes on these incongruent trials, and even when they are correct, they will be slower. They will take longer to make the correct response. So this just gives you a flavor of some of these kinds of tasks, computer tasks that we use. All right, so I want to say a little bit about studying the brain. And I guess many or most people in the audience here have learned a little bit about studying the brain, or maybe you do study the brain yourself. So I'm going to say very broadly, you can categorize brain measurement technologies into two groups. So there's imaging and there's electrophysiology. And this is an oversimplification, but with imaging, the idea is to come up with pictures, to take pictures of the brain. And with electrophysiology, the goal is to measure the electrical activity that's emitted or generated by the brain. So these range in various scales. So you have something like fMRI, where you're measuring the blood flow response to neural activity. This generates these kinds of images, so you're measuring fairly large-scale networks. This is non-invasive, it's done in humans. And then you can do small-scale imaging of individual neurons like this. And then there's electrophysiology. So it's done with EEG or sometimes MEG, done on the head. I don't know who this guy is, but he doesn't look very happy. And you can also do electrophysiology invasively, usually in animals. And in here, so this is a neuron here. This is the tip of the electrode. So you can imagine how tiny the electrode is. Now this distinction is a little bit artificial because from imaging data you also get time series, and from electrophysiology data, and the big advantage of electrophysiology is the incredible temporal precision, so you can measure neural activity at the millisecond timescale. And electrophysiology will also give you images. So it's not a real hard distinction between these two. But one way that I like to think about this distinction between imaging and electrophysiology is if you think about circuits in the brain, regions of the brain as being like radios, then what imaging will tell you is whether these radios are on or off. And with electrophysiology, you get a little bit more detail. So it can be more difficult, depending on how you're doing the electrophysiology, it can be a little bit more difficult to know exactly where these radios are coming from in the brain, where the radio station is. But you get to tune into the time-varying fluctuations in the signal. And this is actually relevant, this is interesting, because we know that information in the brain is encoded in different frequency channels. That's actually reminiscent of light, if you think about the spectrum of light. So here's frequency on the x-axis, here's the visible light spectrum, and then here in this frequency range you have FM and AM radio. If you're not sure what radio is, then the rest of us are getting too old, I guess. Now in the brain, you can also generate a frequency spectrum. It would look something like this. So this whole spectrum is actually even off of this chart here. So these are slower frequencies. You'll see how slow they are in a few minutes. But you see that there are peaks in the spectrum, so that, that is taken as evidence for information in the brain being processed in narrow frequency ranges. And that's what you get with electrophysiology. So how do you get a spectrum that looks like this? And how are we able to extract this frequency-specific information out of a signal? So if you think about this time domain signal, this would be from electrophysiology, from EEG. So the data you get coming out of an electrode sort of goes up and down, it oscillates. These are voltage potential fluctuations. And the idea of frequency analysis is to take a sine wave at some frequency and line up the sine wave with the time domain signal. And then you basically have, you have these two signals, and you want to see how similar they are to each other. So you do something that's like a correlation. It's not exactly a correlation, but it's a bit analogous. So you would say, you know, you can quantify the amount of similarity between this pure sine wave at this frequency and this signal that we measured from the brain. And that level of similarity is plotted on this spectral plot, this power plot, at the frequency corresponding to the frequency of the sine wave. So then you repeat this analysis for lots and lots of different sine waves, and ultimately that allows you to build up a spectrum that looks like this. So then you can say, for this signal, this brain signal, it looks an awful lot like a sine wave at 6 hertz, and it looks a lot less like a sine wave at 20 hertz. So that's sort of the basics of the Fourier transform. Now that's kind of nice, except that this sine wave, this is a pure sine wave, so the amplitude response is constant over time, which means that this gives you a static spectrum. So this tells you about the characteristics of the brain activity over a large time window. But your brain is never static, right? Your brain is constantly doing things. You're constantly getting new sensory input, new memories, new thoughts. So the brain is highly non-stationary. And so this static spectrum is not really sufficient for understanding how the brain is changing in its frequency characteristics over time. So what we do is basically stretch out or decompose this static spectrum into a dynamic spectrum, which is typically called a time-frequency analysis. And here, to read these plots, so this is time on the x-axis, frequency on the y-axis. So here, lower numbers down here correspond to slower frequencies, and higher numbers here towards the top correspond to faster frequencies. And then here you see on the y-axis, the power is getting higher or lower, and here that corresponds to the color. And so in fact, the average over time of this plot is how you get this plot over here. All right, everyone clear about signal analysis 101? So where do errors happen in the brain? When you make a mistake or when you almost make a mistake and you correct yourself at the last moment. From imaging experiments, from fMRI experiments, this comes from a meta-analysis, a review. We can see there's a hotspot here in the medial prefrontal cortex, so I've mentioned that term before. So this is kind of like the radio station that's broadcasting to the rest of the brain that you just made a mistake, or maybe that you're about to make a mistake and you need some corrective action to avoid that mistake. So that's useful information. But we want to know what's happening in this radio. What exactly is the signal that's being broadcast by this radio station here? In other words, what do errors sound like? So if you could kind of listen to these errors, what would it sound like? So it turns out that these errors have a very specific spectral characteristic, a real specific signature that we measure with EEG. That signature is called midfrontal theta, so I'll use that term a few more times. It's called midfrontal because if you look at the topography, the distribution over the scalp, it's right over the midfrontal scalp areas. And if you want to know where that is, the next time you make a mistake, you go like this, right? This is like, why do we do this when you make a mistake? That's exactly the place where we see this error-related signature, response conflict signature. I don't know if that might be coincidental, but. Okay, and it's called theta because over time, over the course of the past century, people have given different names to specific ranges of frequencies in the EEG. And theta is the term for around 6 hertz or so. So 6 cycles per second. So here you see a trace of EEG data, and you see that there's this ongoing theta rhythm. Here's when the stimulus appeared on the screen, and here is the button press. Now in this trial, this is one trial, the research participant actually made the correct response. But it was one of these cases where they could have made an error. They almost made an error. And then you see there's an increase in the amplitude of this ongoing theta rhythm. It begins before the button press is made, and it continues for a few cycles, and then it eventually decays back down to baseline levels. So it's a pretty rapid response. You can see, so this time here is 400 milliseconds, so this whole period out here would be around 1 second or so. So it's quite a fast response. Okay, but how fast is theta? I could say that's like on average 6.5 hertz across people, but what does that mean? So to give you some insight into what theta, so how fast this error-related signature is, I need a research volunteer. So who is going to do some audio? Yes, thank you. So how do you like my talk so far? It's interesting, okay. So I would like you to applaud. Not anyone else, just you. But you have to applaud as quickly as you possibly can. Faster. Great, thank you, perfect. That is theta. The fastest you can possibly clap your hand, that's around 5 or 6 claps per second. And so here is a plot of frequencies, and here you see some of the different names that are given for these frequency ranges. These are very slow frequencies that you see mostly during sleep and deep sleep. And here you have theta here, and then much faster. So this range here from delta up to gamma, these are generally considered like the cognitive frequencies. These are the frequencies of brain rhythms that are most commonly associated with cognitive processes like perception and action and language and memory. And theta is one of the slowest rhythms. So as fast as you can clap is generally considered a pretty slow rhythm for cognitive brain function. So let's see, here you see a time frequency plot. Like what I showed you earlier. And here you see this modulation in between 4 hertz and 7 hertz. And it's fairly brief. It lasts for a few hundred milliseconds, which is a few cycles. And here you see the topography. This is like you're looking down on the top of someone's head. Here's the nose. There's no hair because they always use me as the model for these maps. All right. And what's really remarkable to me is so in this plot here, the data are filtered around theta, so I'm only showing you theta. So you don't know that it's specific just from this plot. But we do these analyses in a very open, very exploratory, data-driven manner. And I don't impose midfrontal theta on the data. I say, let the data, whatever features are in the data, they will just pop out. So we look at every channel everywhere in the scalp. We look at a large range of time, a large range of frequency, and the only thing that really comes through for these situations where there's response conflict and you're about to make a mistake, that's really just midfrontal theta. So I find that quite remarkable. And this is highly replicable now. So replicable, in fact, that it's boring. If you tried to do this research and just publish this basic finding, it won't get published anywhere because it's too much. The editors don't care about it. It's boring now. All right. Let's see. I'll skip that slide. So these kinds of plots reflect averages over hundreds of trials and a few dozen research participants. Usually we try to get 20 to 30 people per experiment to make sure that that the findings are really statistically significant. But this gives you like a bird's eye view. So we want to zoom into this picture a little bit more. So what we do is take advantage of the noise across trials, the variability across trials. And so one trial might look like this. So here you have the trial onset. Here's a stimulus that the subjects have to respond to. At some point later in time, they press a button. And then so we have the timing of the button press, and we also have the brain signal from that trial. So we measure the button press time in milliseconds, and then we measure some feature of the EEG. And then that goes into one dot here. So in this graph, here's the behavior from this trial and the brain signal from this trial. And then we go to trial 2. The reaction time is going to be different. The brain signal is going to be a little bit different. But again, we extract these two measures and put them on a plot here. And we generate this for lots and lots of trials, dozens of trials, hundreds of trials. And then we fit a line through this function here, or through these data. And this line basically tells us about the linear relationship between the behavior and the brain signal over trials. And if this line is positive, it means they're related to each other. And if the line is flat, if it's 0, it means that the brain signal is uncorrelated with the reaction time. So you can probably see where this is going. Here we see, now this is a time frequency map, but this is a time frequency map of correlation coefficients. So we actually run this analysis over lots and lots of different time points and over lots and lots of different frequencies. Now when people are in these kind of boring trials, the congruent trials, you don't really see much of a correlation. But when we look at these mixed correct trials, so remember that was the correct trials that contain this partial error, so this muscle twitch for the incorrect response. Then you see all of a sudden a really strong correlation in midfrontal theta. So midfrontal theta is just sort of popping up all over the place. Again, this is a purely data-driven analysis, so I'm not imposing my a priori expectations on the results. All right. Another experiment. Should we do another experiment? The question is, why is this in theta? What does that mean, that it's midfrontal theta? Why isn't it anywhere else in the brain? And why is it in theta? This is a weird, why there's this pulsation at 6 hertz. And why is it 6 hertz? Why not 2 hertz? Why not 17 hertz or 100 hertz? Okay, so we're going to do another experiment here, audience participation, but everyone gets to do this. You don't have to clap. I mean, you have to clap at the end because it would be really awkward. I'll start crying if you don't clap. So what we're going to do, so everyone make a fist like this. Extend your pointer finger. No, not your middle finger, your pointer finger. Okay, so what you want to do is keep the upper joints fixed and only move the lower joint, so the joint that's connecting your finger to the hand. And move it back and forth like this, so down and up. And you want to take about 2 seconds to get down and about 2 seconds to get back up. And what you want to, and keep some tension on your fingers so you make sure that the finger is really straight. And what you want to do is pay attention to the sensation, the physical sensation coming from your finger. It's a bit of like mindfulness training here. And try to make your finger movement as smooth as you possibly can. And the question is it smooth? Do you feel like your finger is really moving smoothly? The answer is no, it's not moving smoothly. Your muscles are jerking forward like this. It feels like there's some friction in the joint. And it's not because you're getting older, and it's not because you spend too much time using this finger on your phone, although that probably is also true. In fact, these data were from 1992, before they even had smartphones. All right, so the joint angle is relatively smoothly moving. But when you look at the EMG, so the electrical activity coming from the muscle, you see that they're bursting. Your muscles are sort of bursting forward. And this is happening pretty fast. This is 50 milliseconds, so it's a 20th of a second. Here you see the power spectrum of this EMG signal over 8 different individuals, and it has a peak here in upper theta, so around 8 hertz or so. So there's something intrinsic in the muscle physiology, and this is not just one, kooky example. There's something that's built into your muscle physiology that causes fine temporally sequenced actions to happen in the theta range. So your body likes theta. That's a preferred frequency of temporally sequenced actions. So that was one example. Here's another example. This is also from this 130 million keystroke study. Here you see a distribution of the interkey intervals, so basically how long it takes you to get from one key to the next key. And that's also around 150 to 200 milliseconds, which is theta, so that's also around 4 to 5 hertz. Here you see, this is a kind of interesting study where there was like this wiggly line template on the screen, and then there was another wiggly line, and you had to move the mouse. The research participants had to move the mouse to get their wiggly line to match the template wiggly line. And when they looked over time, what people are doing with the mouse movements is they would move the mouse and then hold still and look and compare, and then move the mouse a little bit more and then look and compare, and move the mouse a little bit more. And when you look at the duration of stopping, so how long it takes you to sort of move the mouse and then stop, and then move it again and stop, those durations also had a distribution like this, peaked at around 200 milliseconds. Again, that's theta. So 200 milliseconds means 5 times per second. Here, going back to this partial error data that I showed earlier, when we look at the what we call the correction time here, so this is the time lag between the onset of the muscle, the incorrect muscle, to the onset of the correct muscle. So that's the correction time. And lo and behold, that is also, that peaks in theta. Let's see, I think that was the last example. But this is, so there's many other examples that I could give. Language is also in theta. People utter around 6 syllables per second. And people who say and like a lot, they tend to, those are like, no, I'm doing it. These are meaningless syllables. So people who use these kinds of informationless syllables tend to talk faster, but the rate of information is still around 6 meaningful syllables, 6 chunks of information per second. And giggling is another example. So if if you're tickling someone, or if you were being tickled, then giggling is also in theta, which is pretty interesting. All right, so that doesn't really tell us what theta means or why your medial prefrontal cortex is broadcasting in theta. What is the function of this rhythm? So I think part of the answer is this, that your brain needs to synchronize with the body, and the body has this preferred physiological response in theta, or sort of motor physiological response in theta. The other answer is we really just don't know. What I think might be happening is that there's a phase coding mechanism in the brain. So what does phase coding mean? Phase coding means that there are different computations that are taking place in different phases of the cycle. So here's an oscillation. You have the peak, you have the trough, the rising slope, the falling slope. These are different phases. And so the idea, what I think is happening, is that the circuit in the medial prefrontal cortex, which is monitoring your actions and signaling to the rest of the brain that you are about to make a mistake, that monitoring is happening rhythmically in this pulsed fashion. So there are phases where the brain is monitoring your actions, and then there are phases where it is broadcasting this signal that control is needed. So it's a fairly simple theory, and this general idea of phase coding has a long history in neuroscience, so it doesn't totally come out of nowhere. Turns out this is really difficult to get good evidence for in humans, non-invasively. We are trying, so we have some ways that we've developed to try and find evidence for this idea. It essentially involves running this brain behavior correlation that I explained earlier, but in a phase-restricted manner. So we're looking to see if there are correlations between EEG and reaction time that are unique to specific phases of the ongoing theta rhythm. So that's ongoing work. The preliminary results are positive, so we do see that there's a phase modulation of the brain behavior correlations. But I'm not going to show you all those data right now. Instead, I want to talk about where we go from here. So we started doing this work in 2007, I guess. I think that was still before Twitter, right? Yeah, it's hard to imagine those days. Whenever you're starting something new in science, it's like really exciting at first because there's this new thing that you're discovering and nobody really knows what's going on. And then you quickly get into this sort of boring phase where nobody cares about your research anymore because the exciting stuff is already done. But they still don't really believe that it's a real effect, right? Because there's replication issues, there's noise, there's reproducibility problems. So, you know, you have to go through this boring phase of replicating. You have to see if you can get the same finding, the same pattern of results with different experiments. You have to try with different data analysis methods, different experimental techniques. You have to get other people in different labs to replicate your results, but they don't want to do that because they're already bored, you know. So that took maybe a little bit under a decade, I think, to get past this point of sort of, yeah, I guess I shouldn't keep calling it boring, but sort of we needed to make sure that what we were looking at was a real effect that you could put in a textbook. And I do believe that this is a real effect that you could put in a textbook. This finding we basically see, so we've now looked in hundreds of subjects, and it's basically there for everyone. Sometimes, if you don't see this effect within a single individual, it's usually because the data quality is low for other reasons. Maybe the signal wasn't very good. Or maybe they were a pigeon instead of a human. I'm sure pigeons have the same thing, actually. Okay, but where do we go from here? So then at some point you have to, you're sort of allowed to like break out of this replication phase and start moving into more interesting avenues. So there are several important avenues of future research for midfrontal theta. And conflict monitoring, conflict processing, and error avoidance and learning. One of them is bringing midfrontal theta from these computer tasks, which are pretty artificial, more into real-world, realistic behavior. And this is now much more feasible than it was in the past because of improvements in technology that allow for mobile EEG, wireless EEG recordings, and so on. So my group is not working on this, but other groups are working on this topic. And here you see an example of EEGs. Well, the research participants were playing a computer game, which, for better or worse, is basically real-world behavior nowadays. And they replicate these same findings. So you get strong midfrontal theta when there's a mistake or when you're about to make a mistake, when you get killed by the enemy. Here, so this is one important future direction for midfrontal theta. Another important direction is incorporating midfrontal theta into other cognitive processes, and in particular more high-level cognitive processes, the kinds of cognitive events that you typically think of when you think about sort of brain function, higher brain function. Again, this is not something that my research group is actively working on, but there are other groups who are linking midfrontal theta to sort of like higher-level learning models, for example. Another important line of research, which we're also not doing, but other groups are doing, is bringing this into the clinic and trying to understand both how midfrontal theta might be used as a potential biomarker for various diseases or for predicting treatment outcomes. And also just to, from a more scientific perspective, to see if studying this feature midfrontal theta in patient groups can tell us more about the nature of midfrontal theta because of what we know about those patient groups. So here you see an example looking at depression, so patients with a major depressive disorder and controls, and you see that their error-related theta, midfrontal theta, is stronger than the controls. So it kind of seems like this error signal is being broadcast much more intensely in these individuals compared to the matched controls. So there's another important future direction, and this is the one that we are doing in my group, which is instead of going from midfrontal theta to a higher level cognitive process or real world behavior or clinical contexts, we are actually going down further into the brain, more from a biological and neurophysiological perspective, and trying to understand what is the neural circuitry that underlies midfrontal theta. So when you measure the signal from EEG, you get this, this wobbly kind of signal. And there is something in the brain. There is a circuit in the brain. So these are depicting different types of neurons, 2 different neural circuits. Something is happening in the brain that is producing cognition, the atoms of cognition, and simultaneously generating these electrical fields that we measure with EEG. The problem is that just from measuring EEG, it's really, really hard to know what these circuits look like, what these circuits are doing. Let's see what I have here. So the question is, when you see this, what does this mean? What is happening? What is the brain actually doing? What does a neural circuit look like that produces this response? Because again, this wobbly thing, this time series, this is not this is a measure of brain function, but this is not this is not what implements cognition. Okay, so unfortunately, this kind of research is impossible to do in humans. Also, unfortunately, most Western countries have strong ethics boards that prevent us from doing the experiments that we need to do in humans. But if anyone wants to volunteer, then come talk to me later. It's okay. All right, or if you want to send some of your students, then... All right, so this, I think this is a really important question. I think this is one of the most important questions looking forward into the next few decades in neuroscience. And so I hope I'm not making a mistake by throwing a huge amount of taxpayer money and all of my time and intellectual energy and that of my postdocs and PhD students into trying to make this link. So trying to understand, but it's like Dutch money and European money, so you guys don't have to, if you think I'm making a mistake, it's not your taxpayer money. So anyway. Trying to understand what kind of neural circuit, what kinds of neurons in different layers of the cortex and interactions across different fields within the cortex are actually producing this electrical field. So as I mentioned, this research is not possible to do in humans. So we moved to rodent models. It's also not possible to do in the institute where I work. So we had to build our own lab. And we started that in 2016, so about 3 years ago. Here you see one of my postdocs unboxing some of the first equipment. Here's some of the picture of the lab being just set up. This is from our first pilot recording. And my postdocs assured me that this is what the lab looks like every single day, morning till night. I'm too busy doing administrative stuff to ever really spend much time in the lab, so I just believe them. I hope that's not a mistake. All right, so this is the goal. This is my lab now. Our major goal is to understand the biological, the physiological origin of midfrontal theta by measuring EEG while simultaneously manipulating features of the microcircuitry. And so we can access particular features of the microcircuitry. We can up or down regulate particular molecularly identifiable classes of neurons in different layers of the cortex while simultaneously measuring EEG, and that basically allows us to identify links between what we measure at this macroscopic aggregated scale and what's going on under the hood. So I'm going to show you a few pictures of some of the electrodes that we're using. These are variously for rats or mice. This is 40-channel EEG. It's hard to see from the scale, but this whole thing fits on a mouse skull, which is quite small. So the scale bar got sort of disappeared somehow. I'm sure it wasn't my fault. But it's something like this is a millimeter. So this is this is just a couple millimeters across. It's a tiny thing. For some of the experiments that we wanted to do, the kinds of electrodes that we need, the kinds of recording setups that we need didn't exist. So we had to design some of our own electrodes. Some of them are produced by NeuroNexus, for example. Some of them are produced by Cambridge Neurotech. And some we build ourselves, which that picture is missing also. Certainly not my mistake. So these are the ways that we measure. You can see how small these are. So the measurement basically is all in microns. These are the ways that we measure brain activity with very high fidelity in space and in time. And these are the two ways that we manipulate or can up and down regulate neural activity. We can use electrical microstimulation where we deliver a very small current into the brain that kind of generically activates the brain circuit that's inside the electrical field area of the stimulating electrodes. And we're also using a fairly new technique called optogenetics. How many people have heard of optogenetics or familiar with optogenetics? It's basically a way to up or down regulate the activity of neurons according to their molecular categories. So we can identify molecules that are in specific types of neurons and then cause expression of ion channels that will open or close, which will cause the neuron to become excited or inhibited. And the way that we can cause these ion channels to open or close is by shining a light at a particular frequency into the brain. All right, so by way of summary, You make small mistakes and near mistakes very often, possibly hundreds of times per day. We don't actually know how often people are making these small mistakes because they're hard to measure. But we certainly know that a lot of these are happening unconsciously. So you're not even aware that you're making these mistakes and almost making these mistakes and then correcting yourself at the last moment. So there's noise in the brain at every spatial scale of brain function. And we also engage in this balance between exploring the environment and exploiting the environment. And these are explicit strategies that can generate mistakes, that can generate errors locally, but at a sort of more behaviorally global level, they turn out to be optimal strategies. In the lab, when we measure EEG, we find this very idiosyncratic pattern of EEG. So it's like a signature, it's almost a biomarker of errors and response conflict when you're about to make an error. Understanding exactly where midfrontal theta comes from and exactly what its computational significance is largely unknown. So we have some ideas that we've published, sort of theoretical predictions. And of course, we are currently working on this in my lab, and these are important future research directions. So hundreds of millions of years of evolution has specifically engineered your brain to produce mistakes all the time. So don't feel bad about learning from don't feel bad about making mistakes. You should feel bad about not learning from your mistakes. This one's quite a mouthful, but basically there's synchronization between the theta that we see in the brain and these temporally sequenced motor actions. And we think that there is kind of a cyclic or rhythmic repeated computation of monitoring your behavior and then catalyzing this brain response to adjust your behavior when you're about to make a mistake. Okay, so the future of midfrontal theta is bright, with many open questions that are becoming answerable due to new neuroscience technology, including improvements in invasive and non-invasive electrophysiological measurements, and new techniques for up and down regulating neural activity, including optogenetics and improved electrical microstimulation. So that's it. Of course, I don't do a lot of this, or really any of this work on my own. So I have a really nice research group in my lab in Nijmegen, which is the good pronunciation. I've had several PhD students and postdocs, a full-time technician. We are always looking for more people. In particular, right now, we're looking for postdocs. We have an internal funding mechanism for postdocs who are more than 2 years away from their PhD. So if you're interested in joining my group and you meet those qualifications, please come talk to me or send me an email. And thank you for your attention, and thanks again for bringing me out here.
Okay, thank you, Mike. It was a very interesting talk. Hope you all now know better about how your brain patterns look like when you actually make mistake, and what is midfrontal theta signature is, and actually think about it, how you want to play with it and actually manipulating your mistakes in order to learn better from the mistakes that you do. I think we have around 10 minutes for question and answers, and if you have any question, okay. So let's... Working?
Yeah.
Okay.
For the pilot test, have there been any adverse effects on the test subjects?
You mean in the rodent experiments?
Yeah.
No, so we do the stimulation to be as weak as possible. So the weakest stimulation level that we can give to elicit a neural response. And the thing is, these responses are also very spatially local. So we generally don't see any behavioral responses.
I have another question. In the near future, do you think you will increase the strength of the stimulus?
Yeah, so there's so some people use optogenetics as a way to activate or regulate specific behavioral patterns. And so that's not what we are doing just yet. We're trying to focus a little bit more on the fundamental physiology. So in fact, we are trying to avoid eliciting any specific behavioral pattern because we want to focus more on the brain response. But once we've gotten a better hold on the brain response, yeah, there's all sorts of interesting questions that you can ask, like up or down regulating the medial prefrontal cortex to see if you can induce this kind of experience of conflict or at least the behaviors associated with conflict. Yeah, so check back in 5 years or come join my lab and do that experiment.
Dr. Michael Cohen, thank
Mike
you for
is good,
amazing talk. That was a lot of knowledge we gained today. And URI is thankful to Yalda for bringing the right Michael Cohen here. I have a very simple question. It's so how have you seen any signature of impact of age or gender in your data when you collected a lot of data from a lot of participants, human participants?
Yeah. Yeah, good question. First of all, please call me Mike. Dr. Michael X Cohen is just too long and too formal. Good question. The answer is we haven't looked into that. We could, with gender, certainly, we have enough variability there. With age, we tend to use mainly university students. So I don't think we have enough variability to find an age effect if it's there. I would say there's certainly going to be no change up until the age of 39, because that's how old I am. Once you hit 40, it's just, yeah.
Thank you for the talk. The task you're talking from the beginning was mostly engaging motor task. I mean, some
Yes.
muscle movements, and the mystics you're mentioning and the theta samples are all from, relating to some movement.
Yes.
So what if we change the task to when they're making a decision, they do not move anything, they just, for example, counting something or some cognitive thing?
Yeah, great question. So let me go back, sorry about that, to this slide here. So you can see this is actually quite a big chunk of real estate here in the brain that gets activated in this review over different tasks. Indeed, we focus more on fairly simple motor tasks in my group. That has several advantages. And I like to keep things simple. But you do see that when the nature of the task becomes more cognitive, more abstract, the kind of hotspot, the zone for activating conflict and error-related signaling tends to shift a little bit more anterior. And if it's somehow related to emotional conflict, then it gets even more anterior and a bit more ventral. So there is this gradient. And it's not really clear whether it's the same mechanism, the same computation, just happening in different parts of the cortex because they get different inputs from different other cortical and subcortical areas, or whether there's some commonality in our experience of the conflict, but the actual computation itself is a bit different for these different kinds of conflicts. That's an open question. Yeah.
So I'm thinking about the theta. Is it larger with a mistake?
Yes.
So if you were to take a task, like even your keystroke one, where you get better at it, does the theta does it decrease? Is that what
Yeah.
you see?
So that's a good question. So one of the things that I like about these kinds of tasks is you can't overtrain people. You can train people, you can't overtrain people. So this conflict effect never goes away. And that hasn't gone away in me. And I've done I don't want to know how many tens of thousands of trials of these kinds of tasks. What we do see is that errors in particular, so we can separate the electrophysiological response to errors on, conflict on correct trials. So where you actually made the correct response, but you almost made an error, versus where you actually made an error. And what we see there is that when there's a full-on error, you actually made a mistake, there's this midfrontal theta signature, and then there's also this additional lower frequency delta component, and that you can see this dissociation here.
Yeah, so you can see it in the signal. It's like an almost mistake and a mistake, right?
Yeah. So the thing is, when you make a mistake, you almost make a mistake, and then you actually do make a mistake. Right, so you have these two cognitive processes that get mixed. And in this study, this was like 60-something subjects, and we were able to isolate these two signatures. But you were also asking about learning effects. And they're generally, if you make the conflict less severe, then the effect gets smaller. Right, so if you give people, if you say, you can take as much time as you need to identify the color of the word, then these conflict effects get smaller because there isn't as much conflict. You have to drive the speed pressure a little bit to get these effects to be larger. Yeah.
Thank you very much for the presentation. There is a slide that you had only 2 words on it, exploring and exploiting.
Yeah. That was back here.
Yeah, well, I love these 2 words because I do reinforcement learning, and
Right.
I love these 2. So I have 2 questions. By any chance, are you doing any reinforcement learning? This is the first question. The second question is, which one of these 2 are the most responsible for the making more mistakes?
Yeah. Good question. So the first question is not really anymore. So during my PhD, I was doing quite a bit of reinforcement learning and incorporating some computational modeling, like Rescorla-Wagner style modeling. I haven't done that kind of work in years, a few years, mainly because I've been trying to focus more on the kind of systems biology perspective of midfrontal theta. Which one of these is more associated with errors? I think they both are. If you are just exploiting your environment, then you might be missing a better deal. Right? So if you always go to the same restaurant, you always order the same thing, you could be enjoying other food even more, and you'll never know. So that can be a mistake. I think exploring, but I think the answer to your question is probably exploring is more associated with. This is where you might be missing out on something with exploitation strategy, but you don't necessarily know that you're missing out. Here with exploring, you're much more likely to get negative consequences because you go to a different restaurant, you order a meal that you don't even know what's in it, and the chance of you not liking it is higher.
I'm just interested in knowing, within your college population, in your research, if in regards to the variability that Dr. Kunal was mentioning, but is there variability in the sense, even if it's subtle, are you able to see variability, especially with the motor mechanism?
You mean variability in like the partial errors, for example?
No, well, in general, before a partial error, during a partial error, with an error, and after. Like, is there, like, for that band, is there a lot of variability?
Yeah, so we see quite a bit of variability in basically everything we look at. So there's variability in the behavior, variability in the brain activity. Some of this variability we try to account for with, for example, these correlations with reaction time. So we do see that some of the variability in reaction time is attributable to variability in the theta response. We also see variability in the peak frequency of theta. So some subjects have faster theta, some subjects have slower theta. This is the average over, this was like 60-something subjects. So there is some variability. Some subjects have a faster theta peak, so it's earlier, I mean, some subjects have a theta peak that's later. It's not totally clear what's causing some of these sources of variability. Either we don't know, or it could be just noise. Some of it, I think, is just noise. And other things are going to be, could be genetic factors, and yeah, not sure. But definitely there's a lot of variability.
Yeah. Okay, thank you.
Thank you for the talk, very interesting. Have you done these studies in disease models or on any drug effects?
So my group, the answer is no. Disease models, definitely yes. There are other groups who are working on this with regards to depression. I showed one picture of that. And OCD and Parkinson's disease. Actually, I have been involved in some of the Parkinson's work, but it's not a focus of my group. As for drugs, are you referring to more medications or like recreational?
Like amphetamines, nicotine, drugs that commonly affect executive functioning.
Oh, right. Are you asking that because I live in Amsterdam? Yeah, so in the context, so with dopamine drugs, yes. So dopamine medications in the context of Parkinson's disease. Other medications and recreational drugs, I'm not sure, but I think mostly the answer is no. Yeah.
Thank you.
Yeah.
Last question. John.
First, thank you for the fantastic talk. I just had a quick question about the slide on depression and the differences between control and healthy and that midfrontal theta expression. So I found the differences between the two groups fairly striking. I was wondering if you feel that this change in midfrontal theta is a relatively reliable biomarker of depression, and even further, could downregulation of this response have an effect on some of the symptoms of depression?
Yeah, it's a great question. So I was not involved in this research, so I don't feel comfortable really answering that question with a lot of certainty, but I guess I would just refer you to this. Bismarck and John Allen is the PI from this group. Yeah, I guess an important question is whether this kind of response has sufficient specificity to be able to, look at an EEG in one individual and draw any conclusion about their level of depression. And I don't know the answer to that. So if these are relatively weak effects, then it might be something that, you see them at the group level, but they're not necessarily you can't be so confident about any individual. So yeah, you would just have to look up that paper. Yeah.
Okay, thank you everybody again. So outside there would be a quick snack. Please feel free to help yourself. Dr. Cohen is going to be available here for another 3 hours, so
yes, exactly.
feel
Really
free
3
to
hours.
ask questions.
Okay.
No, joking. Okay, thank you.